Welcome, everyone. We're going to let everyone filter in, and then I'll introduce Dr. Fukuda. So welcome to the people joining. We'll give it a little more time, and then I'll introduce Dr. Fukuda. Okay, I think we're almost up to our normal live participant viewer numbers. So I'm going to go ahead and start my introduction for Dr. Keitaro Fukuda, or Keith, as he's affectionately known. Dr. Fukuda is an MD PhD graduate of Keio University School of Medicine in Japan. He is currently a senior research scientist at the Laboratory for Skin Homeostasis for the Riken Center for Integrative Medical Sciences, or the Riken IMS. He is also an adjunct assistant professor at Keio University School of Medicine in the Department of Dermatology. He actually performed a 3-year postdoctoral fellowship with Dr. John Harris at UMass Medical School, which is how I came to know Keith. We were both in the lab at the same time. And today he's going to be presenting to you a wealth of data that he generated during his time in the laboratory about melanoma immunotherapy research. Dr. Fukuda is also a member of many different societies, including the Japanese Society for Investigative Dermatology. He has many different research grant supports from different institutions in Japan, including the Grant in Aid for Young Scientists from the Ministry of Education, Culture, Sports, Science, and Technology. Keith is also an inventor on a patent application as a result of his work in Dr. Harris's laboratory. So we're very excited about the implications of his work and the translatability to human patients. So with that, I will share my screen and present his recorded talk. And then Dr. Fukuda was kind enough to stay awake for the live Q&A session. So he will be here to answer all of your questions. All right.
Thank you for giving me the opportunity to make a presentation today. My name is Keitaro Fukuda from Department of Dermatology, Keio University School of Medicine, Tokyo, Japan. Today, I would like to talk about the new therapeutic target for melanoma immunotherapy called AIM2, which is a cytosolic DNA sensor that is expressed in dendritic cells. This is my COI disclosure. Melanoma is an aggressive skin cancer that is particularly immunogenic, and it is one of the cancers that show high response rate to immunotherapies such as anti-PD-1 antibody and ACT, adoptive T-cell therapy. However, over half of the patients still doesn't respond to these therapies, and there is a need of new treatment that enhances the efficacy of current immunotherapies for melanoma. Growing evidence supports that the number of CD8 T cells infiltrated into the tumor correlates with the prognosis of immunotherapy. Therefore, the responders to the PD-1 antibody has a high number of CD8 T cells, higher percentage of PD-1 expressing CD8 T cells, and higher percentage of PD-L1 expressing tumor cells compared to the patients who doesn't respond to anti-PD-1 antibody. The CD8 T cell rich tumor is now called as T cell inflamed tumor or hot tumor in the cancer immunology field. It has been reported by the researchers of hot tumor that the spontaneous CD8 T cell infiltration into tumor is facilitated by the STING signaling pathway in the cytosol of tumor infiltrating dendritic cells, TIDCs. The activation of STING signaling pathway leads to production of type 4 interferon and thereby promote T cell priming in the tumor draining lymph node and T cell homing to the tumor. So based on these findings, now a number of pharmaceutical companies developed their STING agonists, and now they are performing the clinical trials against several types of cancers by the treatment of STING agonists plus the anti-PD-1 antibody. So while the importance of CGS STING signaling pathway in the cytosol of tumor infiltrating dendritic cells, TIDCs, are now well established in the cancer immunology field, The cytosolic tumor-derived DNA can also be recognized by a DNA sensor called AIM2, whose function in melanoma remains unclear. AIM2 is a component of AIM2 inflammasome, and it can produce a cytokine, I1 beta and IL-18, and it can also induce a form of the cell death called pyroptosis. So in this study, we examined the functional role of AIM2 signaling within the melanoma microenvironment during immunotherapy. To determine whether the AIM2 is expressed in melanoma and regulates the melanoma progression, we subcutaneous challenge wild type and AIM2 knockout host by B16F10 melanoma B16F10 melanoma cell line. This is a melanoma cell line that doesn't respond to anti-PD-1 antibody, so it is one good example for cold melanoma. We found that the AIM2 knockout host had a significantly lower tumor burden compared to wild type host. When we looked at immune cells infiltrated into tumor by flow cytometry, we found that the number of CD8 T cells and CD4 T cells in the tumor were similar between these two groups. However, the AIM2 knockout mice had a lower proportion of regulatory T cells, Tregs, and the higher CD8 T ratio in the tumor compared to the wild type mice. We also looked the number of macrophages, the dendritic cells, CD103 positive or CD11B positive dendritic cells, and the number of these immune cells infiltrated in tumor were similar between these two groups. We also looked the interferon gamma or TNF alpha producing CD8 T cells, and the percentage of these cells were similar between these two groups. However, when we checked the protein production of interferon beta, I1 beta, and IL-18 in the tumor, we measured by ELISA, and we found that the AIM2 knockout host had a significantly more interferon beta and significantly lower I1 beta and IL-18 protein production compared to the wild type host tumor. Since it has been known that the major interferon beta producing cells in the tumor are the dendritic cells we next wanted to determine whether the AIM2 expressed in the dendritic cells infiltrated into the human melanoma. Therefore, we performed the immunohistochemistry of CD11C and AIM2, and we found that the thicker melanoma, T3 stage melanoma, have a yellow cells, which is AIM2 expressing dendritic cells compared to thin melanoma, T1 stage melanoma. We did this experiment about around 30 samples, and we found that the DC numbers were similar between these thin stage and thicker stage melanoma. But when we look the number of AIM2 expressing dendritic cells and percentage of AIM2 positive DCs, the thicker melanoma had a higher number and higher percentage compared to that of thin stage melanoma. These results suggested that the AIM2 expressing TIDCs are increased in patients with melanoma and correlate with the tumor progression. Since it has been known that the cytosolic DNA activates STING type 1 signaling in antigen-presenting cells, we examined the role of AIM2 on this process by treating mouse bone marrow-derived dendritic cells, BMDCs, by the B16F10 melanoma-derived DNA. We found that the AIM2 knockout BMDCs produces a significantly higher amount of interferon beta and CXCL10 compared to wild-type BMDCs in response to tumor-derived DNA. Intriguingly, these responses were abolished in AIM2 STING double knockout and STING single knockout BMDCs, suggesting that the enhanced production of interferon beta and CXCL10 in AIM2 knockout BMDCs requires STING signaling. Therefore, we check the expression of STING signaling pathway downstream molecules such as TBK1 and IR3. by Western blot analysis. And we found that the AIM2 knockout BMDCs have a stronger expression of phosphorylated TBK1, um, phosphorylated IR3 compared to wild-type BMDCs in response to tumor-derived DNA. And these responses were published in AIM2 STING double knockout and STING single knockout BMDCs. Uh, so collectively, these results suggested that AIM2 inhibition leads to the enhanced activation of STING type 1 interferon signaling in BMDCs in response to tumor-derived DNA. Based on these results, we wanted to test whether the AIM2 knockout BMDCs can be used as a treatment to enhance the efficacy of melanoma immunotherapy. For this purpose, we treated B16 F10 wild-type mice by DC vaccination, which were generated by pulsing the GP100 peptide, and ACT, which is a combination therapy of radiation, IV transfer of GP100 specific CD T cells, also called as PMELs, and IP injection of IL-2. The combination of ACT with AIM2 knockout DC vaccine led to a more robust anti-tumor response than the ACT alone. And among the mice that received ACT with DC vaccine, the mice that received AIM2 knockout DC vaccine showed significantly lower tumor burden compared to the mice that received wild type or AIM2 STING double knockout DC vaccine. When we looked at immune cells infiltrated into the tumor, we found that the mice that received AIM2 knockout DC vaccine treated group had a higher number of PMELs, lower frequency of Tregs, and higher ratio of PMEL Treg ratio compared to the mice that received wild-type DC vaccine. Importantly, this enhanced anti-tumor immunity of AIM2 knockout DC vaccine required STING signaling because the mice that received AIM2 STING double knockout AIM2 STING double knockout DC vaccine treated group showed similar tumor growth curve and similar immune responses as the mice that received wild-type DC vaccine. Since it has been known that the STING type 1 interferon signaling promotes the T cell priming in the lymphoid tissue, we examined whether the AIM2 knockout DC vaccine can enhance the priming of CD T cells. For this purpose, we done the same experiment using this time PMELs that were labeled with CFSC. We found that the PMELs infiltrating in the spleen, the mice that received AIM2 knockout DC vaccine had a higher proliferation index compared to the mice that received wild-type DC vaccine, and also the number of PMELs infiltrated into the spleen AIM2 knockout DC vaccine treated group had a higher number of PMELs compared to wild-type DC vaccine treated group. So these results suggested that the AIM2 knockout DC vaccine has a stronger impact on priming of PMELs in the spleen compared to wild-type DC vaccine. Next, we examined whether the enhanced anti-tumor immunity of AIM2 knockout DC vaccine requires recognition of tumor-derived DNA in melanoma microenvironment. For this purpose, we done ACT plus DC vaccination plus intratumoral injection of DNase1. And we found that the addition of intratumoral injection of DNase1 completely abrogated the enhanced anti-tumor immunity of AIM2 knockout DC vaccine. So these results suggested that the enhanced anti-melanoma immunity of AIM2 knockout DC vaccination depends on the recognition of tumor-derived DNA in the melanoma microenvironment. Next, we examined whether the enhanced anti-tumor immunity of AIM2 knockout DC vaccine requires autologous type 1 interferon signaling and the production of CXCL10. For this purpose, we done the ACT plus DC vaccination treatment using AIM2 FNR double knockout and AIM2 CXCL10 double knockout DC vaccine. We found that the aim2 double knockout DC vaccine treated group completely eliminated the enhanced anti-tumor immunity of aim2 knockout DC vaccine. Therefore, the tumor growth curve was similar as the mice that received wild-type DC vaccine. And similarly, but to a lesser extent, aim2CXCL10 double knockout DC vaccine decreased the enhanced anti-tumor immunity of aim2 knockout DC vaccine, and the tumor growth curve of the aim2CXCL10 double knockout DC treated group showed intermediate between the wild type and aim2 knockout DC vaccine treated group. When we looked the immune cells infiltrated into the tumor by flow cytometry, we found that mice that received aim2 and aim2CXCL10 double knockout DC treated vaccine treated group showed significantly lower number of females compared to the mice that received aim2 knockout DC vaccine. And when we look the Treg number, the mice that received aim2 and aim2CXCL10 double knockout DC vaccine treated group had a lower frequency of Treg compared to the mice that received wild type or aim2 double knockout DC vaccine treated group. So these results suggested that the enhanced anti-tumor immunity of aim2 knockout DC vaccine requires autologous type 1 interferon signaling and promotes the female's infiltration into tumor by a CXCL10 and suppress the Treg infiltration into a tumor by a type 1 interferon signaling, but not by a CXCL10. Next, we examined whether the limitation of I1 beta and IL-18 production leads to the enhanced anti-tumor immunity of aim2 knockout DC vaccine. For this purpose, we done the ACT plus DC vaccination treatment using the I1 beta knockout or IL-18 knockout DC vaccine. Both the I1 beta knockout and IL-18 knockout DC vaccine treated group showed intermediate tumor growth curve between the mice that received wild type and aim2 knockout DC vaccine treated group. When we look the immune cells infiltrated into the tumor, both I1 beta knockout DC and I1-18 knockout DC vaccine treated group showed lower number of females compared to the mice that received aim2 knockout DC vaccine. And when we look at the immune Tregs, the mice that received I1 beta and IL-18 knockout DC vaccine had lower frequency of Tregs compared to the mice that received wild type DC vaccine. So these results suggested that the limitation of production of I1 beta and IL-18 also contributes to the enhanced anti-tumor immunity of aim2 knockout DC vaccine by suppressing the Treg migration into the tumor. Finally, we checked whether the suppression of pyroptosis can also lead to the enhanced anti-tumor immunity of aim2 knockout DC vaccine. So for this purpose, we done ACT plus DC vaccination experiment by using the DC vaccine that are CD45.2 positive, and we used wild type hosts that are CD45.1 positive. And we check the number of DC vaccine infiltrated into the tumor-draining lymph node and spleen. And we found that the DC vaccine infiltrated into the tumor-draining lymph node spleen at day 10 and day 20 after the female's transfer. There were no difference between these two groups. So these results suggested that the enhanced anti-tumor immunity of aim2 knockout DC vaccine doesn't depend on the suppression of pyroptosis. Since it has been known that the failure of the PD-1 antibody treatment is due to the insufficient number of CD8 T cell infiltration into tumor, and also because we found that the aim2 knockout DC vaccine promotes the CD8 T cell infiltration into the tumor, we asked this whether the PD-1 antibody plus aim2 knockout DC vaccine can enhance the efficacy of anti-PD-1 antibody in cold B16F10 melanoma mouse model. So we treated wild type mice bearing B16 if the melanoma by control IgG, PD-1 antibody, wild type or aim to knock out DC vaccine, or a combination therapy using PD-1 antibody plus wild type or aim to knock out DC vaccine. And we found that the PD-1 antibody plus aim to knock out DC vaccine treated group was the only group that showed significantly lower tumor burden compared to the mice that received control IgG. And when we looked at immune cells infiltrated into the tumor, the result reflected the result of tumor growth curve. So the PD-1 antibody plus aim to knock out DC vaccine treated group was the only group that showed higher number of CD8 T cells, lower number of Tregs, a lower frequency of Tregs, and higher CD8 T ratio in the tumor compared to the mice that received control IgG. Collectively, these results suggested that the aim to knock out DC vaccination provides active anti-tumor effects when combined with anti-PD-1 immunotherapy. Finally, we address whether the therapeutic implications in mouse melanoma model can be extended to human immune system. So for this purpose, we treated human monocyte-derived disease by control or M2 siRNA and stimulated by human melanoma-derived DNA. We found that the human MODCs that were treated with M2 siRNA had enhancement of STING type 1 interferon signaling. So therefore, there was a stronger band in phosphorylated IRS3 in M2 siRNA treated MODCs than the control siRNA treated human MODCs in response to tumor-derived DNA. And we also checked the interferon beta and CXCL10 production. And we found that the AIM2 siRNA treated human MODCs produces significantly higher amount of these interferon beta, CXCL10, compared to control siRNA treated human MODCs in response to tumor-derived DNA. In addition, we found that the M2 siRNA treated human MODCs produces significantly lower amount of IL-1 beta and IL-18 in response to tumor-derived DNA compared to control siRNA human MODCs. So these results suggested that AIM2 in human MODCs responds in a similar way to mouse BMDCs and thus can be used to generate a DC vaccine to improve the efficacy of melanoma immunotherapy. So in summary, AIM2 plays an immunosuppressive role in melanoma microenvironment. And if we eliminate AIM2 during DC vaccination in combination with ACT or anti-PD-1 immunotherapy, that will lead to the limitation of IL-1 beta and IL-18 production, and also it enhances the STING type 1 interferon signaling pathway. And this will lead to the promotion of PMLs, migration into tumor, and suppression of Tregs into the tumor, and thereby convert cold melanoma into hot tumor. So targeting AIM2 in DC vaccine is a novel therapeutic strategy to enhance melanoma immunotherapeutic responses. In the end, I would like to acknowledge all the members in the Harris Lab and also the people that helped me a lot of experiment of this work, especially Kate Fitzgerald, Anastasia, and Luban Lab members. And also I would like to acknowledge the member in Keio University School of Medicine, Dr. Funakoshi, Kawakami, and Yaguchi. Thank you for your kind attention.
That was a great seminar. Thank you so much, Keith, for recording that and for joining us for the Q&A session.
You're welcome.
So we have no open questions yet. I actually had a couple questions. How do you think this could be best used in clinic? Like, do you think the siRNA approach is the best, or do you think that we could do like a small molecule, or how do you think we can achieve this?
Oh, yes. So actually, when we look for tumor cells, the AIM2 role is sometimes beneficial if we block the AIM2 in some type of cancers, but there are some cancers that if we inhibit AIM2, then that will promote the progression. So it is better to just block the AIM2 in dendritic cells. And so I think the best way is to kind of deliver siRNA to dendritic cells only, not to the tumor cells. I think that is safer for the all the cancer treatment.
Makes total sense. What do you think about the mode of delivery? Like, do you think it's better to treat them, ex vivo and reintroduce them as a vaccine, maybe autologous, like, from the same patient? Or do you think there's a way that we could target the siRNA, like through delivery to the actual dendritic cells that are infiltrating the tumor?
Oh, thank you for that question. That was actually asked by the reviewer. Monocyte-derived DC is the kind of popular dendritic cell population that are used to create DC vaccine in the past. But now many data shows that it is better to use the conventional DC that are actually infiltrated into the tumor. So I think it's better to just deliver the siRNA to the dendritic cells infiltrated in tumor. And that is also convenient for the therapy. It will take much longer time to develop DC vaccine by ex vivo. So convenient way and also higher efficacy can be expected by delivering siRNA.
Yes, that makes total sense. Do they, would you envision that the dendritic cells would be isolated like after a tumor debulking surgery and then just, treated with siRNA and then used as a vaccine, or like, logistics? I'm just thinking like how this would be implemented.
Yes, that is kind of difficult question to answer at this moment. Some people think that it is better to just keep the immune system as much as possible. So like to use the DC vaccine as a neoadjuvant setting. If we remove the bulk tumor, then that may have a chance to decrease the amount of dendritic cells also infiltrating tumor. So maybe doing the biopsy, and if we can find there are decent amount of dendritic cells, then that may better to perform the siRNA treatment before the therapy. I think that will be case by case.
Makes sense. Thank you so much. I also wanted to mention that Keith's work was just recently accepted to the Journal of Experimental Medicine. So congratulations on that.
Thank you.
So we have a question from Ann Rothstein. Ann says, hi, Keith. I think you mentioned increased type 2 interferon in the AIM2 knockout mice. Is that correct? And how does that fit into the model?
Yes. So because when I look the interferon gamma expression by each cell, each cell has like similar amount of interferon gamma production, but because that AIM2 knockout DC vaccine promotes CD8 T cell migration into the tumor, the total interferon gamma amount is much higher in the AIM2 knockout DC vaccine treated group. And interferon gamma usually upregulates the AIM2 expression. So I think tumor has kind of defense system to escape from the interferon gamma attack. But if we block that AIM2, and then that will be able to promote the CXCL10 production and also limit the IL-1 beta and IL-18 production. And that is the kind of reason that AIM2 inhibition works for the tumor immunotherapy.
Thank you. That was a beautiful answer. I feel like we end up thinking about that a lot too, with like, migration of cells too. It's just a question of like, is there a lot of migration because there's like a lot of chemokine there, or is it like, you know, that you have these like foci of cells that are pumping it out? You know, is it like a numbers game, which it sounds like you think it's a numbers game, like just the number of cells that are there to make interferon gamma, rather than just like on a per cell basis, like how much they're pumping out. So it's definitely something that I like to think about too with immunology. We have another question from Chi. He says, great talk, Keith. Do you think it's possible to model metastatic melanoma in a mouse for testing potential treatments?
Yes, so I think not only the local stage melanoma, also I think all the dendritic cells infiltrated into the tumor can have a chance to use the AIM2 for to prevent the immune system. So if the metastatic melanoma also has dendritic cells infiltration, I think if we block that AIM2, that will also enhance the anti-tumor response of those satellite metastasis lesion, just as like primary melanoma.
And I actually don't know this. How often do the Mets also have, like if the primary tumor was hot, do the Mets also become hot, or are they sometimes cold? Like, do you know like how often that happens?
Yes, so I don't know much about the difference between metastatic and primary lesion, but many reports say that beta catenin activation or like P10 loss that usually can be found in metastatic lesion leads to the development of cold tumor. So it's better to deliver the dendritic cells to those lesions because sometimes cold tumor doesn't have the infiltration of dendritic cells. In those cases, it might be better to deliver DC, sRNA target, AIM2 sRNA treated DC vaccine, just as I done with this mouse model. If we use the sRNA delivery system only, then if there is no DC infiltration, that doesn't work for those cases. So it's also kind of difficult to estimate whether those metastatic lesion has the DC infiltration or not. But there are some reports that if there is some kind of mutation, there is a high chance of few DCs infiltrating the tumor.
So it would also go with mutation burden then too.
Yes, yes. So sometimes maybe DC vaccine deliver is better than the siRNA treatment.
Makes sense. That's awesome. I think we don't have any more questions in the chat. But that was beautiful work and such big implications for patient management and treatment. So thank you so much, Dr.
Thank you
for your work,
you.
for being with us today. It was a wonderful, wonderful talk.
Thank you very much.
Yeah, and thank you to all our viewers for joining us for Dr. Fukuda's talk, and we'll be back to meet next month for another Global Derm Talk seminar. So thanks, everybody. Bye.
Bye.